Hello, thank you for the opportunity to talk about genetic variation in human models of atopic dermatitis. Here is my conflict of interest declaration. And here's an outline of what I hope to cover in the next 10 minutes or so. So we're going to talk about human AD models in vivo, and specifically what we can learn from genetic studies. I'll highlight some selected examples, as well as briefly discussing the strengths and limitations of this approach to understanding atopic dermatitis. So human AD models fall into two major categories. We can study the genetics, including individual variation contributing to this complex trait, as well as rare severe phenotypes that are often monogenic diseases. In vivo studies also include human models in vivo, and these will be covered in Killian's talk later in the session. So focusing on genetic studies. We know that eczema is a very diverse disease, both in the presentation on individual patients and also the disease associations and trajectories of disease throughout life. In keeping with this very diverse trait, genome-wide association studies have shown multiple risk loci across the whole genome, encompassing genetic functions that make sense in terms of what we know about skin barrier dysfunction, as well as innate and acquired immunity. Together, GWAS studies have identified approximately 31 loci in the white European populations, accounting for up to 20% of the heritability of this complex trait. Latent class analysis is a statistical approach to investigate disease subtypes. And in atopic dermatitis, longitudinal latent class analysis has demonstrated that there are indeed distinct subsets within atopic dermatitis. And interestingly, these distinct subtypes do have different genetic risk profiles. Specifically, the most early onset, persistent and severe subtypes have the highest burden of genetic risk. So moving on to monogenic diseases. These have revealed new insights into atopic pathophysiology, of which loss-of-function mutations in the filaggrin gene are the best known and best studied example. So taking the phenotype of a child with severe early onset atopic dermatitis, up to 50% of children with this phenotype will have a filaggrin loss-of-function mutation, either 1 or 2 loss-of-function mutations, and of these children, more than 1 in 10 will have severe lifelong atopic dermatitis, often associated with other atopic or non-atopic comorbidities. But even in the children that don't have a loss-of-function mutation, we've shown that many of them have a lower copy number within the repetitive filaggrin gene. And putting aside these genetic variants, it's probable that really the majority of patients with atopic dermatitis have a reduced filaggrin expression at protein level, irrespective of their genetic predisposition, because we know that atopic inflammation itself reduces filaggrin expression. So this monogenic disease, ichthyosis vulgaris, has highlighted the central role of skin barrier dysfunction in atopic inflammation. But there are many other monogenic diseases that have also added to our understanding of atopic disease. In the category of skin barrier dysfunction, in addition to filaggrin, loss-of-function mutations in cornea desmosine cause appealing skin syndrome, and variants in this interferon receptor gene cause a subset of atopic dermatitis associated with eczema herpeticum. Multisystem atopic inflammation can occur in the lungs, the bloodstream, as well as the skin. And examples here include SPINK5 mutations causing Netherton syndrome. Barrier dysfunction in the bowel can be caused by desmogliin or desmoplakin mutations, resulting in severe dermatitis, multiple allergies, and metabolic wasting. And lastly, there are many immunodeficiency syndromes that have some features of atopic eczema or other atopic phenotypes, and several examples are listed here. So what are the strengths and limitations of using genetic models to try and understand a complex disease like atopic dermatitis? So one of the major strengths of studying genetic disease is that it gives insight into novel biology. And because our genetic makeup is inherited at birth, by definition, this must predate the onset of disease. So we can be more confident that it's having a causal effect rather than just an observational association. And one of the most obvious examples of the novel biology that we've learned about in AD is the emphasis of the importance of skin barrier dysfunction as a predisposing factor for multisystem atopic disease. Another strength of studying atopic disease genetics is that it gives us indications for targeted therapy. And an example here would be the JAK-STAT pathway, which can now be targeted with JAK inhibitors to treat atopic dermatitis. Limitations of using genetic diseases to try and understand AD is that obviously not all manifestations of monogenic disease are relevant to the more common atopic dermatitis that we see in clinic. Another limitation is that AD is really more diverse and more complex than any of the monogenic diseases that are being studied. So to summarise, genetic models for AD are important in that they've provided novel insight into understanding aspects of AD pathophysiology. They provide opportunities for treatment repositioning, as well as, in the future, potential specific targets for aspects of AD physiology. But it's important to remember that really much further work is needed to fully understand the complexity of AD that we see in our patients. Thank you very much for listening.